What's up? It's me, and it is January 5th, 2022. New Year's off to a great start. And yesterday I made a video where I was talking about the basics of antibodies and just starting off with the antibody structure, the Y shape, the composition of heavy chain and light chain subunits, and how they kind of are designed to do many different things in the body, right? So I guess today what I'm going to be talking about a little more is about antibody activity as it's related to the different cells which produce them, right? So when you hear about antibodies, you hear a lot about B lymphocytes. Okay, so I was talking about yesterday how essentially what happens is that a T helper cell, a helper T cell that's activated by a macrophage, right? The activation occurs through the macrophage's production of interleukin 1, IL-1, which is the primary signaling cytokine that a macrophage will use to activate helper cells, helper T cells. Once those helper T cells are activated, and remember this activation occurs because an antigen is present for the macrophage. A pathogen is consumed by the macrophage, presents an antigen on its major histocompatibility 2 complex, and the T cell then the helper T cell, which is also known as a CD4 cell, then detects the presence of this antigen on this MHC2 complex, and then attaches via one of its receptors, and then gets activated by this interleukin. Then the T cell, helper T cell, essentially activated helper T cell clones itself and then finds B lymphocytes and T lymphocytes, other T cells, right, undifferentiated T cells and B cells, and it then allows them to go through the process of their role in the immune system, which in this case we'll talk about the B cell role, because the T cell role, the T cells are differentiated into killer T cells later down, or cytotoxic T cells later down the line, but that's a different branch. And cytotoxic T cells have a more direct killing effect on pathogens, but the B cells are essentially the step in the production of antibodies that needs to be talked about. So what happens, the B cell gets activated by the T cell, helper T cell, right? The helper T cell, and this is just another thing I need to note, is that these helper T cells, there's many different kinds of helper T cells, right? There's the main helper T cells, then there's there's other kinds that do other things down the chain of your system, but essentially they work all in respect to the humoral immunity system. So this is where the humoral immunity essentially starts, is that the mature B cell then starts its antigen activated by the T cell. It produces antibodies, right? It'll produce plasma cells. So what'll happen is a plasma cell is essentially a cell that has a lot of endoplasmic reticulum. It's essentially, some have called it the weapons factory of the immune system, because the plasma cell produced by the B cell will launch missiles, antibody missiles. And remember, the antibody missiles have several effects on pathogens, okay? So we'll go back into that in a second, but the interesting part here is that the plasma cell then is designed to combat specific pathogens, right? So later on, after an infection is truncated, or is mitigated by antibodies and other parts of the immune system and phagocytosis and things like that, then the plasma cells are no longer have an effect anymore, right? So what will happen is that the B cells will they're no longer needed. So the B cells, some of them will be recycled, etc. But the B cells will produce what's called also a memory B cell, which are long-lived cells. So a memory B cell is a cell which can turn into a plasma cell that is designed to produce antibodies for that infection that was already defeated. So basically, if the infection ever comes back, the same type of infection, the memory B cell kind of stores the memory of how to defeat that infection and will produce plasma cells that will produce those antibodies as they did in the first incarnation of that infection, or the first introduction of that infection to the immune system. So yeah, basically it's like a hash map if you're into computer science, right? And your antibody design pattern is stored in this map, and they're kind of released whenever they're needed, right? So in that way, you don't have to have unnecessary plasma cells living in your system all the time. You can have them activated down this chain of plasma cell production from the memory cells as needed by a reintroduction of the infection. So that's, I guess the evolution has found that as a very natural way to evolve our humoral immunity, right? So so I think that's pretty much what I wanted to kind of talk about, really. So I guess we'll be talking more about antibodies and the way they work later on. It's really interesting. So a brief introduction is antibodies have several different ways, more than four, several different ways of stopping an infection. They can agglutinate an infection, which means they can cause pathogens that are stuck by these antibodies to kind of clump together and thus reduce their motility and allow them to be phagocytosed easily. They can just slow down cells, pathogenic cells, so that the pathogenic cells can never get to their target tissue or their food source, and thus they die. And then they can activate the complement system, which we talked about before. They can activate killer cells. They can do all sorts of other secondary activations to allow phagocytosis to occur through opsonizing a cell, the same way as the CD3 proteins opsonize cells as well, right? Meaning they tag pathogens for destruction by macrophages. So essentially, like we talked about, the overlap of different functions of the immune system. So that's essentially another thing antibodies do. So we'll go more into that later, but that was a quick video, and thanks for your time.